In Essential Task 10.2, we are going to describe the sensory process of vision. For most, I will provide a diagram of the eye, which will correspond to our concept map, similar to how we diagrammed the neuron within Essential Task back in Unit 8. If you do not have that diagram and cannot print it out at home, you can easily use a horizontal piece of paper. As you can see, this is a very horizontally aligned concept map. I will now allow you to pause the video before proceeding into the concept map. Starting at the very top is a foundational term related to sensation, but it is a term that we're going to particularly focus on today. It's the term called transduction. Transduction is the idea of converting sensory stimulations into electrical signals that our brain can interpret. So, for example, our sense of vision, the main topic of today, is going to involve light reflected off objects or things in our environment, eventually turning into neural impulses that our brain can interpret. And that's essentially the very simple two-step process. Our eyes pick up light in the environment. That information, that energy, is then transferred into an electrical signal. We call those action potentials, going back to Unit 8, or neural impulses that are eventually sent out into our occipital lobe in the back of our head, and specifically our visual cortex, for processing. That entire 2 or 3 step process is what we call transduction. Today, obviously, we're going to focus on visual transduction, the process and the anatomical structures involved in taking light in our environment and transferring it into information that our brain can interpret. We'll start on the left under anatomy. There looks like there are 6 columns, though the 5th column in has 3 individual boxes below it. That is the bulk of our concept map, though there are a few boxes on the right side under color and detail. We'll start with anatomy. We'll try to keep the anatomy as straightforward. Unfortunately, a lot of different parts to the eye. As a result, I'll throw you some mnemonics. Hopefully that will help differentiate some of these regions, but nevertheless, we'll start with the cornea. In the diagram, the cornea is the furthest portion to the left here. It's the outside region of the eye. The cornea's primary function is to help focus light in the environment. So, for example, the light that's currently being reflected off the colors on the Prezi right now onto the retina. We'll talk about what the retina is in just a little bit. Arguably one of the most important parts of the eye. It's the part of the eye that had those 3 secondary boxes. So we'll talk about the retina quite a bit in today's essential task. As a mnemonic, I see the word corn in cornea, and I think of corn muffins. It's almost like your corneas are 2 side-by-side corn muffin tops, because we're talking about the outer circular protective layer of the eye, that outer layer. Proceeding onward, we'll talk about the pupil. The pupil is the black portion of the eye, the adjustable opening where light enters. In the diagram, you can see that clearly illustrated here. Your pupil adjusts depending on the amount of light in the environment. So, for example, if it's really bright, you might typically notice the pupil is really small, the black part of the eye. When it's really dark, well, we obviously need to get light into our eye to trigger our retina. As a result, our pupil can then open if there's not a lot of lighting in the room. You could actually very easily see changes in the pupil if you were to turn the lights off and turn them back on. If anybody has any pets, for example, cats, you can very easily see that their pupils can change in size quite dramatically. If it's really bright in the room, their pupils are probably very small, assuming they're not hunting or playing. If it's really dark, it's going to be the total opposite. Humans are exactly the same way. The pupil is the adjustable black portion of the eye. It adjusts in order to let light enter into the eye. In some cases, there's a lot of light in the environment, so the pupil doesn't need to be very big. In other cases, the total opposite. When we look at the word pupil, I see that PU, and I think of the word pew, and I always think of, oh, pew, that doesn't smell great. And I always think of a skunk, a black skunk, and the black pupil. I always connect the PU, or the PEU, if you're thinking about pew, to the connection with a skunk, the black part of the eye. You can make that connection as an easy mnemonic. Continuing onward, we have the iris. The iris is the colored part around the pupil. The pupil is controlled by the iris. The iris controls how big the pupil is. So it is in and of itself what responds to the light intensity in the room. So if it's really bright in the room, it's going to adjust the pupil into a smaller size. If it's really dark, it'll do the total opposite. And it is the colored part of the eye. So whether it's brown or blue or green, each individual person might have a slightly different color iris. As a mnemonic, when I look at the word iris, it almost looks like the word Irish. It's 1 letter off, of course. Whenever I think of the word Irish or the Irish in general, I always think of the color green. Green's not the most common colored eye, of course, but nevertheless, it is a color of an iris that you could very easily encounter. As a result, when I look at the word iris, I think of green, and that's how I always connect this to the colored portion of the eye, which is a really useful mnemonic. If you have a diagram of the eye and they're clearly talking about an internal structure, even if you have varying degrees of familiarity with all of these terms, you can clearly tell that no, it's obviously not the colored part, it's not the iris. You can clearly differentiate this from inner regions of the eye, for example. So we have three mnemonics: your corn muffin tops, your skunk in the middle, the pupil, and then the green iris for the Irish. Starting to go inward into the eye and starting talking about anatomical structures that you can't readily see, the next one is the lens. Right behind the pupil, as light enters into the eye, it bounces off of an anatomical structure called the lens. The lens can adjust through a process called accommodation in order to focus the light onto the retina. Whenever I use the word light, I'm really talking about the details of what you're looking at. So, for example, when I look at this little red dinosaur that hangs out on my desk, when I'm talking about light, it might not seem like it because it's not shiny and it's not bright, but the red tint in the color of this dinosaur is producing light that is currently entering into my eye. It's specifically entering in through my pupil, adjusted by the iris, depending on how much light is actually going on in my room right now. Well, the lens right behind the pupil is taking the fine details of this dinosaur and focusing it onto a region of the eye that we're going to talk about in just a moment called the retina. So it can change shape. The lens can kind of adjust almost like a lens of a camera can. There are some times that you might be looking at something with your camera and it's really blurry, and then you adjust it accordingly with the lens. As a result, the image is much more clear and crisp. That's essentially what your lens does for the fine details of what it is that you might be looking at. As a mnemonic here, in addition to just connecting a lens with what a camera does, I look at the word accommodation, and to connect accommodation to this individual entity of the lens, I always think of the word comedy show. You might take a camera to a comedy show, and it almost looks like a comedy at the beginning of accommodation. Zooming it back out just to kind of put into context where we are and how much is left, we're now going to transition to the retina, which has a secondary level below it. And there's one more region called the optic nerve that you can see before we talk about color and detail. The retina is the region of the eye that I would easily say the College Board asks about more than any other. The retina, or structures within the retina, as you'll see in just a few moments. The retina is the inside portion of the eye. So when we look at a diagram of the eye, it's this back layer, assuming that the diagram that we're looking at is the interior of the eye. The light that enters through our pupil bounces off the lens and then is focused onto the retina. We'll talk a little bit about why that's so important in a few moments. As you'll see, the retina contains what are called rods and cones, and we'll talk about those in the upcoming boxes. As a mnemonic to connect that the retina is the region of the eye, the inner surface that contains the rods and cones, I see the word tin, and I always think about an actual item, a tin item, and I think about things like rods and cones, and maybe rods and cones could be made of tin. You could very easily, as I just did here, Google images of rods and cones that look like they're made out of tin. So I see the word tin in retina. That's how I always connect the rods and the cones. So zooming back out under retina, there are two to the left under the category name of receptors. The rods and cones on the inner surface of our retina have two very specific jobs. Starting with the rods. If you were to look at a picture of the retina, as is indicated here in the bottom left, the rods are on the periphery, or the outside of your retina. As a result, rods are responsible for peripheral vision. Peripheral vision is vision off to your sides. So if you were to extend your arms out perfectly to the side and wave them as I am, it is your rods in the peripheral portion of your retina that are being activated by that sensory input and then eventually sending that information up to the brain for processing. Your rods are also responsible for twilight vision, or vision in the dark. Obviously, humans don't have nearly as strong night vision as other animals, but the night vision that we do have is facilitated by our rods, which actually interesting is that because the rods are on the periphery of our eyes as opposed to straight ahead, at night, let's say you're walking around your room and it's pitch black, chances are, let's say you're looking for your phone, you don't remember where it is, you don't want to turn a light on because it would disturb you. If you're staring directly down at where you're looking, you're actually going to have a somewhat difficult time detecting where your phone is. If you tilt your eyes up a little bit and rely more on your peripheral vision, you'll actually get a little bit of a better image of what it is that you're looking at, whether that's walking around your room or kind of fishing around your items. It's because your rods are stimulated at night during twilight vision or night vision, and they're in the periphery of your retina in the first place. Beyond that, they also detect black, white, and gray. So whether it's black in the environment, white in the environment, gray in the environment, or peripheral/night vision, those are all unique responsibilities of your rods. Additionally, if you were to use your peripheral vision, so I'm thinking you're staring straight ahead and I'm talking about items directly to your right and left, the reason that they look darker than they might actually be, the reason why if you hold an item perfectly off to your side, let's say a 90 degree from where you are here, chances are you're not going to be able to tell the color. It's just going to kind of look dark It's because your rods detect black, white, and gray as opposed to color, which we'll talk about in a few moments with our second type of receptor in the retina. The cones, on the other hand, are directly smack dab in the middle of the retina and are responsible for fine color and detail. This region of your retina, so again, this circular diagram here is sort of a 2D visual of what the back of your eye looks like, the retina. The exact middle part with all these little colors, the red, the green, and the blue, is known as your fovea. The fovea represents the smack dab middle portion of your retina, and that's where the vast majority of your cones are. Your cones are active in generally well-lit conditions, so my room is illuminated, my cones are active, and they are responsible for, as the name sort of suggests here, and some of the discussion that we're having suggests, it's responsible for color and fine detail. If you were to look straight ahead at just about anything, whether it's an item in your room, an item in my classroom, or anything at all, the fact that you can read, for example, this slide, like, for example, if you stare at the word cones, you can read the word cones, but when I see the word cones and I'm staring at it, all of the other text is getting a little blurry. Like, for example, in the bottom left corner, you might notice the word gray from our previous slide, but you can't really read that if your eyes are focused on the word cones. That perfect, crisp, high acuity vision that you're seeing right now, or using when you're looking at the word cones at the top of this box, is because the majority of your cones are smack dab in the middle of your retina in a region called the fovea. As a mnemonic, I see the CO in color and I see the CO in cones, and I've always connected those as a mnemonic. Can very easily differentiate rods and cones. One of the most common questions on the AP exam asks you to do just that. To carry on this a little bit further, I want to talk more about the fovea, because the fovea is very interesting. The fovea, as I mentioned a moment ago, is this cluster of cones smack dab in the middle of your retina. It is your perfect straight ahead vision, and there are a ton of different ways to illustrate your fovea relative to other regions of your eye. As I mentioned a moment ago, when you stare at the word fovea, or like you did a few moments ago with cones, some of the other words surrounding it might get a little bit blurry. You might have a difficult time, for example, reading the terms here in foveal vision if you're only looking at the word fovea. That's because when you directly look at something, the cones in your fovea are lined up perfectly with that entity, whether it's a word, whether it's a sign while driving, whether it's a football in the middle of a game, a baseball in the middle of a game, whatever it happens to be. And that is the element of your field of vision that has the highest degree of acuity and fine detail. We call that foveal vision, that straight ahead vision with the highest degree of sharpness and accuracy, that perfect visual of what it is that you're looking at. When I look at the word fovea, I see the FOV at the beginning, and I think of the phrase full of vision. That's how I kind of always envision foveal vision. It's the part of your line of sight that has the most acuity and detail. Because don't get me wrong, if you look off into the distance, yeah, you can see a variety of things off into the distance. But what's directly straight ahead, perfectly lined up with your fovea, is what you see in the most detail. If you're trying to read something from across a room, you'll absolutely be able to read what it is that you're staring at, but things off to the sides get a little bit blurry. And you have to shift your focus from one entity to the other to actually change what it is that you can fully read. And that's because every time you do so, you're lining up your foveal vision, this direct line shot from the fovea to that entity, bit by bit every time. Fovea is a little bit interesting. We'll talk a little bit more about it in this upcoming portion here over to the right. The last anatomical structure is called your optic nerve. What's really fascinating is if you look at a diagram of the eye, you might notice that there is this literal giant nerve that exits out of the eye right here. That is what we call the optic nerve. When the rods and cones are stimulated in the retina, they have the responsibility of telling the brain the information that it is that you're looking at, whether it's the fine details of this dinosaur, whether it's a sign across the room, or whether it's a book that you're reading. The rods and cones stimulate nearby bipolar and ganglion cells, which combine to form what's called your optic nerve, particularly the ganglion cells. It's essentially just a bunch of neurons that exit out of the eye, as you can see here in the image. They have the responsibility of sending the action potentials to the brain about what it is that you're looking at. Action potential is a term we talked about in Unit 8. It's a synonym or a term that we use for describing neural impulses or a neuron firing, a neuron sending information. Well, your optic nerve has the responsibility of taking all of the detail of what you're looking at, the color, the shape, and all of that, sending it to the brain. The reason we're talking about this is that technically creates a very small portion of your retina where there are no rods and cones because there is literally a hole for the exit of the optic nerve. It produces this interesting phenomenon called a blind spot, where there is technically a portion of your field of vision at any given moment where no information is being sent to your retina, and therefore no information is being sent to your brain. And as a result, your brain actually just kind of fills in a portion of your field of vision at any given moment with what would, generally speaking, be there. And you never really notice it. It's not something that you notice at all day in and day out. You really have to do something like the demo that I'm about to describe in order to actually acknowledge the fact that we have a blind spot in the first place. But the fact that we have a blind spot is a byproduct of the fact that there needs to be nerves exiting out of the eye so the information can be sent up to the brain about what it is that you're looking at. This might require you to pause your video. I've also posted information about how to do these demonstrations on Google Classroom. So it's going to be hard to kind of describe here. You'd have to really pause the video and like move around your computer. It might work best, honestly, if you open up the links in Google Classroom on your phone. But you can actually detect the presence of your blind spot by carrying out the description here in this demo. The final part, though, of this essential task on vision and visual sensation is to talk about the color and detail. So these are exclusive to this portion of the essential task. These are sort of isolated from anatomy, but they are theories pertaining to why we detect color in the way that we do. The first leading theory is called the Young-Helmholtz trichromatic color theory. This can sometimes be called the trichromatic color theory as well, so be wary about that possible synonym. The trichromatic color theory states that our cones, again, the portion in our retina that are receptive to color in fine detail and well-lit conditions typically, come in three different types: red, green, and blue. And that, almost like mixing paint, these three cones combine together in order to produce all of the different colors that we see. So our ability to see purple and yellow and orange and every other possible color is a byproduct of differing levels of each of these cones being stimulated. You might be looking at something that's really red, so there's obviously a lot of red cones that are active. You might see something that's more of a purple, so it would be a combination of maybe a little bit of the red cones and the blue cones together, maybe not so much green. And that every different color that we see is basically an adjustment of the three different types of cones in our retina. One of the leading pieces of evidence that suggests that the trichromatic color theory is in fact true are examples of individuals with color deficient vision. We sometimes call this color blindness. That term color blindness, if you've never really looked into it, is a little bit of a confusing term, because you hear that and you think somebody must not have any color vision. It must just be black and gray, black and white, etc. It's actually not the case. Typically, what color deficient vision looks like is there being one of the three cones that is in some type of deficiency. And as a result, the individual has a difficult time differentiating colors from one another, as opposed to living in an environment that's just black and white. So, for example, if you look at this visual here, by and large, you should be able to see the number 74, assuming there is no presence of color deficient vision. There's all these little red dots, but there's also the green dots of the number 74. If you can see that perfectly, you do not have color deficient vision. If you look at this and it's all one giant circle of different colors, it would be possible that we're looking at a case of color deficient vision, and that's precisely how we gauge this. One of the most common examples of color deficient vision is a difficulty differentiating between red and green. So an individual with color deficient vision would look at this visual and it would just be a smattering of one color the whole way through. There wouldn't be that clear differentiation between the green and the red. It's interesting, actually, a few years ago, there was a Thursday night football game between the New York Jets and the Buffalo Bills. The Jets wore a vibrant green uniform and the Bills wore a vibrant red uniform. And there was a lot of complaints by NFL fans with color deficient vision or color blindness who could not tell the difference between the two teams on the field because each of the two jerseys were such contrasts in red and green. It's not like one was slight green or slight red. I mean, it was entirely red versus entirely green. And when you have that totally polarized red versus green, an individual with color deficient vision, assuming it is a red-green deficiency, will have a difficult time telling them the difference, if honestly having no ability to whatsoever in some cases. Proceeding onward, there's another interesting theory regarding our cones and our vision. It's called the opponent process theory. The opponent process theory argues that instead of just having three cones with three colors, we actually have three sets of cones that are paired with a secondary color. It's unlikely that the College Board would ask you the amount of detail of actually knowing the pairings, but I would copy it down. They did technically ask a question or two over the years where that was important, but the pairings are red-green, yellow-blue, and black and white, and that these pairings together work in combination to produce the variety of colors that we see. One of the best ways of identifying the possibility of this theory is through the evidence known as after images, or the after image effect. There's this interesting phenomenon where it seems to be the case that if you stimulate the cones for one of those color pairings long enough, meaning you essentially stare at something. After enough time passes, it seems like you overwhelm the cones associated with that color, and as a result, those cones instead send the brain information about the opposite color instead of the color that's actually being detected. So, for example, if you stare at something that's red long enough, your brain might start to send information about the color green, although that's totally incorrect, because the red stimulation has been so overwhelming and so prolonged. The red portion of your cones have basically shut down or turned off momentarily, but they still want to send the brain information about what it is that you're looking at, and now instead send the opposite opposing color, thus the opponent process theory. We can try and simulate this. This works a little bit better in person, but we can try it over YouTube. What I'd like you to do for, let's say, about the next 20 seconds until I say stop is stare directly at the middle of this American flag. I know the American flag has a really unusual color scheme here, but I just want you to stare at it until I say stop. In a few moments, I want you to continue staring as I give these directions. I'm going to ask you to quickly move your eyes from the American flag over to the white portion of the screen. So the white portion where there's absolutely nothing. All right, you can do it. If this worked properly, and it doesn't work perfectly for everybody, obviously we're doing this through YouTube, but for many people, when they shifted their eyes from this American flag over to the right, they actually saw an American flag with the correct color scheme, red, white, and blue, or at least the framework and outline of an American flag. The color portion might not always work, but some people, when they do this demo, actually see the full American flag with the entire correct color scheme. Why might that be the case? Well, if you look at an American flag, the portion here at the top left corner is really blue. Well, what's the pairing for yellow? It's blue. What's the pairing for this color here? And this one's almost like a greenish blue, so it's not a perfect green, but the stripes are typically red, and then the stripes are typically white. Well, what's the pairing for black? It's white. What just happened, and this might seem a little weird, but what may have happened for some of you is by staring at this flag, you overstimulated your yellow, you overstimulated your green, and you overstimulated your black. As a result, your brain is still interpreting information that's being sent from your cones, but your cones, instead of sending yellow, green, and black, sent your brain the opposite color attached to each of the opposing pairs because you overwhelmed the first one. So instead you got red, where the stripes are, blue around the stars, and then white for either the stars or the stripes. This phenomenon is called the after image effect, and there are literally an endless number of these. If this was fascinating to you or it didn't work for you, you could easily Google after image effect and go through a ton of different examples of this. And this works a little bit better in class anyway. But nevertheless, we call that the opponent process theory, and the best evidence for this theory is the after image effect. The very last box is to acknowledge a term related to our sense of vision, discovered by Hubel and Wiesel, called the feature detectors. It seems to be the case that the optic nerves, the nerves exiting our eye, particularly the ganglion cells, connect and send information to specific parts of our brain that seem to be responsible for unique features of vision. Vision is an interesting sense because there's recognition of movement, there's edges, there's shading, there's outline, there's all of this fine detail related to our sense of vision. And it seems to be the case that there are feature detectors in our brain responsible for fine, minute information about what it is that we're looking at, like the shading and the color and the recognition of movement. And it seems to be the case that you could theoretically damage a part of your brain and not lose all of your vision per se. I mean, you absolutely could, but you could lose some ability to detect some of the unique features related to the sense of vision. It's a unique element of vision that's not quite exactly like all of our other senses, but we call these unique specialized cells in the brain. The word cell is a term that people use a lot, but that's another term for neuron. But there are these specialized feature detectors in our brain whose responsibility is to process very particular parts of the sense of vision. Movement, edging, lines, angles, outlines, things along those lines. So the process of visual interpretation, when we get all of this information about what we're looking at, it starts with light in the environment. Light reflected off of anything from this little toy dinosaur to the words on the screen to the words on the screen that you're looking at right now as opposed to the one in class. That light enters in through our pupil and hits our retina, triggers either the rods or cones, or probably honestly a little bit of both. That light information needs to be transferred into something our brain can interpret through the process of transduction. Rods and cones stimulate these bipolar and ganglion cells to send that information through the optic nerve out of our eye and eventually up into our brain, into our occipital lobe in the back of our head for visual processing. We have noted over the years that there seems to be specific receptors, or specific rather neurons, in our occipital lobe that detect fine details pertaining to our sense of vision, angles, outlines, movement, etc.